Good morning, everyone. A very warm welcome to all our distinguished guests, researchers, industrial professionals, faculty members, students, and participants for joining us today. On behalf of Institute of Biological, Biochemical, and Pharmaceutical Sciences, Dow University of Health Sciences, I would like to thank you for taking the time to join us for today in the webinar, Opportunities to Optimize Your Return on Investment, ROI, on Bioequivalence Study. My name is 𝐊𝐞𝐲 𝐓𝐚𝐤, and I am serving as a moderator for today's session. I work as a senior clinical research associate at IBBPS, Dow University of Health Sciences, where I am involved in the management of and coordination of clinical research trials and clinical research projects. So we are delighted to have you. We are delighted to have you with us today for what promises to be an engaging and informative discussion on strategies to maximize value and efficiency in bioequivalence studies. So there are some housekeeping notes before starting this webinar. All participants will remain muted throughout the webinar to ensure there is no distortion in the audio and to improve the audio quality. If you have any questions during the session, please submit them using the chat box. We will address as many questions as I can during the question answer sessions at the end of the presentation. Thank you for your cooperation. Without further delay, let's begin the session today. So now I'm introducing our esteemed speaker. So it is my great pleasure and privilege to introduce our distinguished speaker for today, Mr. 𝐊𝐞𝐲. Mr. Kenneth is currently serving as a senior manager business development at 𝐭𝐭�𝐰𝐴𝐞𝐻. He is a pharmacist by training. He has begun his professional journey with 𝐭𝐭�𝐼𝐴𝐞𝐻 in 2001 as a study coordinator. Over the years, he has played a crucial role in clinical trial management, business development, and regulatory compliance. With more than 2 decades of experience in clinical research and regulatory affairs, Kenneth has managed over 250 bioequivalence studies as an ICH GCP certified professional and has also served as a principal investigator since 2019. His experience also includes leading the COVID-19 vaccine clinical trials. Additionally, he has served as an adjunct senior clinical lecturer in the Royal College of Surgeons in Ireland and University College Dublin, Malaysia campus. Today, he will share his expertise, practical experience, and strategic recommendation on optimizing return on investment in bioequivalence studies. Mr. 𝐊𝐞𝐲, I'm thanking you for joining us today.
Thank you very much.
The virtual floor is now yours. You can start your presentation. Thank you.
Thank you, Chaida. Thank you also to Yunma for organizing this webinar. We've been speaking about this webinar for a while now, and I'm glad that we finally have the opportunity to meet your faculty members. Yeah, Chaida, would you be controlling the slides on your side? Chaida, may I know if you would be controlling the slides from your end?
Yeah, I'm controlling. No, you can control the slides from your end also. If you have the slides, you can also control from your end.
Okay.
You can share your screen. We can see them on the screen. If you have encountered any problem, then I can share my screen as well.
Sure. Then I'll do the slides on my side. But just a very brief introduction. Thank you everyone for taking your time for this webinar. I think bioequivalence is not a foreign word right now, especially in this regulatory landscape. Yeah. So let me know if the slides are seen, Chaida.
और मेथाफॉल और इन लोगों का मेरे पास
I'm requesting the participants to kindly mute their mics. I'm kindly requesting all the participants to kindly हेलो यार, स्पीकर की को क्यों म्यूट करती है? स्पीकर क्यों तो अनम्यूट करो, मैं अपने पास से नहीं कर पा रही हूँ। ठीक है, मैं अपने पास से नहीं कर पा रही हूँ। Hello, Kenneth, can you please unmute yourself? Because we are unable to hear you.
Oh, I'm so sorry.
Thank you
Yeah.
so much. I thought
It's
it's some kind of distortion from my side, but
Okay, slides are okay.
yeah,
Alright.
yeah.
So I was just
You're
saying,
audible now.
yeah, I was just saying that the industry has very much been driven by guidelines from Europe, guidelines from America in terms of how we plan our bioequivalence studies. And as more and more of your pharma industry at the moment are exploring export markets, export to stringent regulatory agencies, you are now thinking about how you're going to plan your bioequivalence, how you're going to fulfill these guidelines, right? Unfortunately, the bad news is you can't have one size fits all. Right? That's a bit of a bad news, right? Ideally, as a pharma industry, as regulators, right, you would like to do one bioequivalence study, and that study would last you for many years. That study would allow you to register your product in as many countries as possible. That's ideal for management. For any pharma manufacturer, that's your goal. In the past, we have big challenges whereby the US have their own guidelines, the EU have their own guidelines, and different parts of the world also have their own guidelines. ASEAN, for example, have their ASEAN guidelines. So a bit of a good news here right now is that we have finally adopted, the world has finally adopted and introduced the ICH M13A, which is the guidelines, ICH guidelines for bioequivalence. It's fairly new. It was implemented in 2025. Many countries are slowly adopting and following the ICH M13A. Okay. If your developmental plan has always been following the European guidelines, the good news is that ICH M13A is very similar to the European guidelines. All right. It's those who have been developing according to the US FDA guidelines, and they have to rethink their strategies. If you look at the US FDA, they always publish the product specific guidelines, the PSGs. And as of last year, they have been revising all their PSGs, and they will continue to revise their PSGs, their product specific guidelines, to provide guidance on what kind of study designs are required for which kind of molecules. I think a very stark difference is really the fat and fasting study. But in the past, right, you've been thinking and planning that, oh, I need a fasting study, I need a fat study, right? Which should I do? Do I do both? Do I do one? Because it's all cost, right? It's all involved a lot of time and cost, and also risk of failing a fasting study or failing a fat study. But today, the ICH M13 guidelines spells out what needs to be done. All right. This. Okay. So the M13 is actually a series of guidelines. What have been implemented today is the 13A. It has been implemented, right? It's already implemented. 13B is already in technical draft. Okay? If you're interested to look ahead, you can actually go to ICH and download the technical draft documents and see what are the discussion points. These are specifically in relation to additional strengths and also the topic of biowaivers. The biowaivers are hot because if you are able to do biowaiver, theoretically, you do not have to invest in bioequivalence studies. So that's forward planning. If you want to think about your different dosage, your different strengths, your different innovation that you have, you may already want to start looking at what are the discussion points in 13B, right? And 13C will start soon. It's a matter of harmonization. 13C elements are already there. There are already previous guidelines which spoke about highly variable products. There are already previous guidelines which spoke about drugs which are narrow therapeutic index and those who are very complex in the study. There were already guidelines. Okay, so it's a matter of a harmonization and making it in ICH. So it shouldn't go too far away from what has already been in place. Yeah. So if we are today planning a study of a product which is highly variable, if you use current guidelines and do your planning, you shouldn't be too far off. Right? You shouldn't be too far off. Similarly, for drugs which are in the narrow therapeutic index group. So this is another snapshot on the guidelines and the implementation. As I mentioned, 13A has already been implemented. 13B will come very soon. The world is waiting for these guidelines, and 13C will follow suit later on. So what is it? Why is it so exciting to have this 13A? I mean, for many people who have been developing drugs and doing bioequivalence studies, 13A means quite a bit for them, right? Previously, different regulatory agencies have different recommendations, right? For their study designs in terms of their population, the type of, even the BMI, right? As simple as unifying the body mass index, what sort of criteria to support approvals, they may have slight differences. And that makes it very difficult for a pharma company when you're planning a study. You'll be a very general question which most CROs like us, they would always ask you when you come to us for bioequivalence study. Same as you go to IBBPS, you say that you want to do a bioequivalence study. I think Yunma and Shida would ask you a fundamental question. Where do you intend to market your product? Where do you intend to register that product? Because decisions for the study design need to be thought about in terms of what is your target market? Right? We would then have to rely on the regulations of the target market to plan the study for you, right? So with ICH M13A, right, the pharma now have got a unified harmonized guidelines, whereby in theory, you would have similar guidelines for various parts of the world, right? I'm still putting it as in theory because there are still some challenges which you need to think about, all right? So for immediate release IR products in the past, when you search, right, when you come to us as CROs, all right, you said you have a molecule to do a bioequivalence study. Okay, the best thing for us to fall on is that to look at the FDA guidelines because they are very transparent. They're very forward in the information. They publish all their PSGs on your website. And you will be able to generally decide whether you need a fasting study, you need a fat study, or you need both fasting and fat, right? Other regulatory agencies like the EMA, European medical agencies, have also started doing their bioequivalence recommendations for product specific, but the list of products is very limited, right? They give a priority on different products and products which are probably more problematic or recently off patent, they will provide a guideline. Okay. One of the challenge, I think, a very stark difference between the European guidelines and the FDA guidelines are on certain products which are so-called in the oncology class, all right, whereby they are not cytotoxic, but they are used in the treatment of oncologies. Your kinase inhibitors, for example, classical example, right? US FDA guidelines requires you to do studies in patient population, which means in cancer patients when you're doing a kinase inhibitor. Whereas if you look at the European guidelines based on the risk assessment and safety, many times they allow some of these compounds to be done in healthy volunteers, right? So it's a big challenge, right? In terms of your investment, you're going to have a oncology product which you have already developed, maybe in your country, in some countries, already registered, already selling, and you're trying to move this product into a market whereby it's more regulated, right? You are stuck with the problems that, well, should I do a study in healthy volunteers or should I do a study in patient population? So that's a big issue, right? Because doing a study in oncology patients is very different from doing in healthy volunteers. But then again, there's the safety aspects to look into, right? So they have different guidance leading to different product-specific guidelines recommendations. You end up needing to do multiple bioequivalence studies, which increase your investment, you increase the time to your market, right? So what has happened, right? The major differences, I just like to highlight some. I strongly recommend that you get yourself a copy of the ICH M13A if you haven't already gotten it. It's available to download. Download it, look for these major differences. I'm going to highlight the major differences to you. And this becomes opportunities, what I mean. Opportunities for you to increase your return on investment, right? The first big question that we always ask, and you would ask IBBPS too, do I need to do a fasting study or a fat study? Right? Even in the European guidelines in the past, right? In the past, you look at FDA, you search the compound, go to the PSG, most likely they said fasting and fat study, right? Then you look at the European guidelines. European guidelines in the past speaks about you need to refer to the approved package insert of the reference product, whether there's any food effect, whether there's any effect on the pharmacokinetics if the study was done in a fat state. Yeah. But in this new ICH M13A, right, section 215, right, a new concept is being introduced, and it's something where for you as a manufacturer or as a researcher, this is the first step you need to think about whether your product, your investigation product, falls under a category of high risk or non-high risk. And that's very important. That's right at the top of the decision tree. When you have a project to think about, the first question you ask yourself is my product considered a high-risk product or a non-high risk product? High risk here refers to the risk of being bioequivalent. That's what the ICH guideline says, is the risk of being inequivalent, right? If you go through the guidelines, they will actually spell out what are some of the areas, right, which makes a product to fall under the category of high risk, okay? For example, if you've got a very specific technology involved, they brought in the concept of not just the product's variability, but they also talk about the process of manufacture. This is a very key difference between ICH M13A versus the previous guidelines. They now even consider the process of manufacture, whether the process of manufacture introduces risk in bioequivalence. Right? In the past, we just look at ISCVs, the intra-subject coefficient variation, to see that is it a highly variable product or is it not a highly variable product, which is an inert characteristics of the API. All right. But today, we also need to think about, because of advances in technology, does your manufacturing process have a process which may cause a risk of bioinequivalence? Or if you're using certain platforms, right? Maybe using certain osmosis systems, you're using certain nano systems in your production of your product, okay? The next key difference in section 2.1.5 about fasting and fat, right? In the past, when we say that, oh, if we need a fat study, design, there will be a standard meal. What kind of meal, what kind of breakfast is provided? It's spelled out in the US guideline, right? The typical American breakfast, high fat, high calorie, right? And they have a sample meal which is provided in the guidelines, what it means by a high fat, high calorie, right? High fat meaning that 50% of the calorie content must be made out of fat. It's a very difficult, challenging meal to eat, right? So that was the old guideline. If you need a fat study design, you have no choice. You have to use a high calorie and high fat. But in the latest guideline, the ICH M13A, they are becoming more scientific and more rational, especially for products which are low risk or called non-high risk products. Should you need to do a fat study design, they actually say that you can actually use a normal calorie, low calorie, and normal fat. The rationale here is very scientific because they say that because most of the time, patients would have a normal meal when they take their medication. Okay, you're not expected to have a very high calorie, high fat meal when you take medication. Because the product falls under the category of non-high risk. Okay, the food effect is not known to cause bioequivalence, right? Therefore, they actually allow different meal types. So that's something interesting, right? For you to plan, for you to think about, discuss with your CROs when you're planning a fat study design. Okay, and these so-called criteria are applicable for your SUPAC applications also. Okay. The next interesting thing is they finally put it in a guideline. I think many people are very happy and excited, and this really helps in your investment. When you have multiple comparator products, when you have multiple test products, right? In the past, it's a guessing game. Can I do a 3-way study? Can I do a 4-way study, right? Would the regulatory agency accept it if I have a European comparator, an American comparator, an ASEAN comparator, right? There's so many different comparator products, right? Today, the guidelines actually spell out that, yes, you can do it, right? If you define your objective very well in the study design, you're actually allowed to have multiple comparator. Okay. What I mean is this: you have one generic product, right? And you intend to sell this, market this in ASEAN. You intend to market this probably in Europe. You intend to market this also in African countries, for example. All right. Your international business team, your global regulatory affairs will need to then check with those countries which is the accepted comparator product. Sometimes it would be different. The European comparator, American comparator, ASEAN comparator could be different. Do you do 3 BE studies, right? In the past, maybe you would have to do 3. But in this current guideline, it actually allows you to do 1 BE study with different reference product done together, right? As long as you pre-plan it, okay, and you report the data accordingly, you can. Okay. Similarly, there's also an interesting component which allows you to do multiple test products. And this again is important when you look upstream to save time. Okay. At times when you're an innovative generic company, right, you may have multiple formulations of a new product, right? You're chasing the patent cliff, right? And as the patent cliff is there, we may not have all the access to all the information of what the comparator product is and it's made of. You may have different formulations of your generic, okay? But you do not know which is the best formulation. You have formulation 1, formulation 2, formulation 3. You set them on stability studies, right? How are you going to decide which formula is the best formula, right? So sometimes doing multiple tests versus a single comparator can be done. In the past, there are a lot of questions. With the current guidelines, it actually allows you to do that. If you have multiple formulations, you want to do a single study in order to save time, you can do that, right? Next, some not so good news. All right, the first 2 items are basically good news to the generic industry who is doing bioequivalence study. This one is not so good news, right? They've introduced a new concept of pH dependency products. What is meant here in this guideline is that some products have got pH dependent dissolutions, right? Or absorptions also, right? In such instances, right, the regulatory agencies are now thinking the guideline wasn't very clear. ICH M13A still haven't given a very clear roadmap of what to do, but you need to plan ahead. Right? You need to plan ahead. Should you have any projects or any compounds which has a pH dependency in terms of absorption, okay, you need to start thinking of your strategies. Okay, what is meant by pH dependency products are that, for example, if the gastric pH is different, right, and that significantly affects the absorption of the product, okay, the thought process is that you may need to do an additional BE study whereby you simulate a condition whereby the healthy volunteers' stomachs are in a different pH condition. Okay. How it's done, the guidelines didn't spell it out clearly, but the American PSG has given one example, right? And then how they do it is that you have to provide a proton pump inhibitor, all right, pantoprazole, right, or one of those proton pump inhibitors to the subjects for a steady state, so that they would have a different pH in their stomach. Then you administer the test product. Okay, so that's an additional study to change the pH of the stomach before you administer the product. It's a bit tricky. We hope that we don't have to do so much of that. Probably the good news is that not so many products are really having this pH dependency issue. Very, very little. Less than 1% of products will have this problem. Yeah. The next two sections, major differences in M13A. Again, these are opportunities, right? They have in section 2.2.1.1, right, now spell out the rules on how you can remove data due to low exposure. Okay. In the past, guidelines and all, removal of data is a very sensitive issue, right? Because regulatory agencies always feel that if you remove data, that you introduce biasness. Okay. But they understand and appreciate that sometimes, right, due to outliers, due to reasons from the product itself, they may be a very low exposure by some subjects, right? They have spelled out the situations where you can remove the data. So do look at this section 2.2 and look into it. What you want to do in terms of your study design planning to ensure that you do not get into a failure problem is that when you write the protocols, when you design your BE study protocols, especially in the statistics section, okay, pay attention to section 2.2 of the ICH M13. Write it well. You allow yourself an opportunity that should the data is not good for those reasons allowed, right? You are able to remove the data from statistical analysis, and hopefully with that, you actually get better BE results. Yeah. And finally, in ICH M13, there's a section which speaks about potency correction. This again is a very interesting and very encouraging change we have in M13A, right? Especially you look at the previous guidelines, in all guidelines, they spoke about the potency, whereby the test and the comparator product should not differ from each other from 5% in terms of the assay of content, right? But I understand sometimes in some countries, right, it's very difficult for you to purchase reference product. It's not so readily available. And when sometimes you get a reference product, the assay of the reference product may differ more than 5% from your product. And that will delay so much timelines in terms of procuring a new batch, redoing your CDPs, redoing your assays. You waste a lot of time and effort. Okay. So the M13A actually today allows you to do potency correction. And you are able to justify to say that that's the batch, right? We are more than 5% difference. When you do statistics, do potency correction, and that helps the statistics to be in a better form. Okay, so these 2 points down there really can save your BE study from failing. Okay, there are additional discussions. In M13A. They actually now recommend male and female. Okay. So there were a lot of questions. Sometimes people ask, do we need female in the bioequivalence study, or can we have all males? Now there's a formal recommendation of you should have male and female subjects in the BE study, provided it's safe for both the population. But the question you may bring forward is that, what's the ratio? Do we need 50-50? All right. The guidelines doesn't spell out any ratio. It doesn't say 60-40, 70-30, or whatever. It doesn't spell out anything. It just says that it's recommended to have a representation of both male and female in your BE study design. And they've also widened the BMI. I think this is in trend with global weight, right? So they've widened the BMI. They've also spoken about very clear guidelines, what to assay in your BE study, whether you're looking at the parent compound or the metabolite, right? What are justifications and what happens to prodrug? Very clear discussions have now been put in this ICH M13A. They also have a very short section about different dosage forms, which are the immediate release dosage forms. Should you have an oral disintegrating tablet, or you have a chewable tablet or suspensions, there is a short discussion on what kind of study, how you administer the product is provided in this guideline. Okay, I quickly jump to a brief introduction of infokinetics. Now we've had now still building our collaboration with IBBPS. We hope that it builds into a good collaboration. In the past 2-3 years, we've been having a lot of conversations. For us, infokinetics, we are about 25 years in operation. We are very glad that our Malaysian regulatory system is in the PICS system, and they have these reference countries where they fall back on the regulatory standards. Yeah. Of course, Malaysia believes that clinical trials do bring in a lot of GNI, right, in gross national income to the country, and a lot of policies are put in place to assist Malaysia to do more clinical trials, right? As a country, the country sets the key performance index for clinical trials. They believe that clinical trials can generate income, can generate skilled workers, skilled jobs, can bring in innovative treatments into Malaysia. Right? We're just going to be very quick about this. Right? Being a 25-year-old CRO, we are very fortunate that we have been inspected by many regulatory agencies. We've been inspected by, in our early years, we've been inspected by the European agencies. We've got the French, we've got the UK authorities, we've also got the Australian authorities. Our later inspections were more US-based. Because our clients started to market their products in the US FDA, filing in both NDA and ANDA applications, and that triggered inspections from the US FDA. So far, we've already been inspected 6 times by the US FDA. For us, we are part of a global hospital group called the IHH Healthcare. They are the second largest healthcare group provider in the world. And we are very fortunate that our BE center is located within their hospitals here in Malaysia. Okay. For us, in terms of quality, we do believe very strongly in maintaining qualities and chasing accreditations. Right from the very beginning of our inception of the company, we have already obtained our ISOs, both 9000 and the 17025. And very shortly after that, we started our GLP accreditations. We've been inspected by various agencies, and we slowly migrate and kept all our ISOs going year after year. What we believe in working with clients is to create value partnerships. We have served a lot of clients around the globe, both from ASEAN, from Europe, and from US. We have served just very few Pakistani companies. We hope that with the collaboration with IBBPS, we can actually provide our services to more Pakistani pharmaceutical companies. So we have only worked with GETS and Pacific so far. Yeah. Our clients have told us what they have done, and their data, their BE reports have gone around all around the world. Right? We are very proud that the work that we do have gone to most parts of the world. There are still some parts of the world whereby our clients haven't submitted their BE reports, especially like South America, right? Whereby you need ANVISA. At the moment, we are not doing any work for ANVISA. So if the client says that they want to seek market authorization in Brazil, we have to be very upfront and say that we may not be able to assist you with the BE study because we are not ANVISA at the moment. But for other parts of the world, we are very happy if you're able to provide you the service and the consultation to do your BE studies. Okay. So maybe in the last 2-3 slides, I'm sure you may want to go into some questions instead. We have in this group various different categories of managers and executives or directors, VPs. How do you then protect your investments? Doing bioequivalence studies put in a lot of money, time, right? I think a lot more time. A lot of time goes into it, and time is very much tied into your investments. Okay, so I just want to give a very brief concept of how can you protect your investments in terms of bioequivalence studies. Yeah, is that you look at this 4C model, look into competency, right? Look into cost effectiveness, look into what sort of customer relationships you can form, and the most important thing, what sort of connectivity that you are able to leverage on. Because ultimately, you have done all you can do, right, in terms of your R&D, in your pipeline development, right? You've got a great product. What's needed is that final bridge, right? That one bioequivalence study report which bridge you to a market access. Okay, so you want to see what sort of connectivity. How can you maximize that work which you have in order to reach wider markets, right? So in terms of competency, I think it's very important on both ends, right? On both ends, and on your end, you need definitely a dedicated team, right? We have seen in most of inquiries which come, right, in our dealings with quite a number of Pakistani pharma companies and also with other companies around the world. Usually, the inquiries comes from the international business development. All right, they are the ones who reach out. They are the ones who reach out. Oh, we need to do a bioequivalence study, or we have these 2 or 3 products or 5 products, right? Yes, although it's international business development who's going to look at it, right, to expand and to decide. But we feel that it's very important that within the organization, you need to have a division. It could be from your R&D, it could be from your regulatory affairs. It depends how you structure your organization. But some organization even call it clinical affairs, right? So maybe I would throw this question to you and ask how many of you may or have a clinical affairs division within your company, in your organization chart. Definitely you have R&D, you have QC, you have QA, you have got business and all, but do you have clinical affairs department, right? To be a great generic company, I think maybe yes, you may want to have a clinical affairs department. Okay, and some of the key roles played by this clinical affairs department is the competency to review protocols, to design protocols side by side with your CRO. Okay? So if you don't have your own resource, you are fully dependent on the CRO to provide you all the information and consultancy, right? So you will ask, okay, I have product A, tell me what to do. So you just have to listen to CRO and they'll tell you, oh, you do this, 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 right? Okay, but you cannot validate it, you cannot verify it. Is that true? All right. So I feel, through the 20-odd years of doing bioequivalence study, it's important, right? Personally, when we are in communication with a team where they have clinical affairs, right, the discussion of study design is very good because we speak science. But when your discussions are with international business development, it's also good because you speak business, but it's challenging because sometimes there are the technical aspects which is hard for international business development to think about. Yeah. Some examples. There are special protocols which are not written in guidelines, right? And this needs experience, right? So the competency of the CRO or the BE centers which you are selecting to work with, right, should have the experience or the know-how, or at least SOPs on how they manage difficult compounds, right? Difficult compounds, right? There would be some compounds which cause vomiting, even in healthy volunteers, nausea, right? I'm sure you know the compounds. You look at the package insert, you know, right? The probability. There are some compounds which has got a very high potential for adverse events, right? Cardiovascular safety, endocrinology safety. So how are these managed? Because if these are not managed well, you end up having dropouts. And when you have dropouts, that's when your statistical problem comes in. Okay, so special protocol designs, how to manage them. These are where you really test the competency of your team or the CRO who's going to manage your products. Okay. And you may also have compounds which are under controlled substances. Okay, these again are niche. They're very niche because you need special import permits, export permits, special protocols on how to manage controlled substances. So if you have such projects, it should be on your checklist. You're in your vendor qualification checklist. You want to check and see, can the CRO, can the BE center manage controlled substances, right? And finally, sometimes some protocols are not just PK endpoints. Some protocols require pharmacodynamic endpoints. Have the CROs, like the BE centers, have they had the experience in doing such projects? So in order to protect your investment, you need to ensure that the people, the PIs, the team who's going to do the project for you have the correct level of competency, right? You may have a preferred vendor, but sometimes for certain projects, you may want to choose certain CROs which are specialized to do those kind of projects. Next would be really customer relationships. This is really entirely up to your organization. Like for us, we feel that there are those big, huge CROs, right? And there are the smaller CROs like IBBPS, like InfoKinetics. We call ourselves small CROs. We're very small in size. We're not global. We don't have a global footprint. Yeah, there are those big CROs which are everywhere around the world, right? So it depends. What sort of relationship that you can form with the CROs, right? Sometimes it's the small CROs, like which are more attentive, who will provide you more customized services, who will listen to you a bit more, right? But at the end of the day, you need to be aware because the project that you do is going to last a long time in terms of the potential for market authorization, right? 5 years down the line, you may want to file a market authorization in a new country, right? And that at that time, you need support from the CRO to provide you documentation. Are they still there? Are they still in existence, right? So these are the aspects you need to think about continuity, right? To get continual support, right? Filing a dossier 5 years later, the regulatory agency may ask you a different set of questions altogether. So if your internal team is unable to technically answer them, you need to get back to the CRO who did the study for you 5 years ago, right? We believe that small CROs are more flexible because they make decisions much easier. They have a very thin top executive. They can react to fast decision making. And of course, cost is very important, right? Cost is very important. We know that India, China produces the world's finished product and API, right? They have a lot of advantages, right? They may have low costs and they have a lot of BE centers even in China and India also, all right? But to find really competent BE centers, they are very high in demand, right? And they're really good CROs in China and India. They don't come cheap also, right? They're also very high in their cost. Okay. I think bottom line is that when you're doing a project, you need to see how fast you can get the project started, right? Certain countries may be very good, but it may take a long time to get the project started because of import permits and export permits or MOCs and many other aspects here. At this juncture, I feel that it's important that every country needs their own BE center. Right? And I've been over and over speaking to IBBPS about it. I feel that Pakistan needs your own BE center to be able to provide you that kind of immediate projects, right? At times to do even pilot studies within your own country, to do formulation selection, right? To do mini projects before you move it to a pivotal study, and you need it fast. And things which are done within your own country, timelines can be managed much better. Yeah. And finally, something for you to consider will be connectivity, right? Connectivity, this would then be a bit of an advantage of the CROs and BE centers when you work with the out of your country, because they get exposed to a lot of other SOPs, right? The CROs which do what sort of client base that they work with? Are they just doing work for generic companies, or are they also managing clinical trials for the regular pharma companies, like the global pharma companies? And you find that if they are doing global trials, right, they would have an edge in getting the more recent regulations and trend, yeah, and that's when you have latest SOPs being utilized, and those kind of knowledge would be then transferred to a regular bioequivalence study. Okay, and sometimes the country that you work with, they're also familiar with multiple regulatory requirements. Okay? For instance, InfoKinetics have worked with so many different clients for different countries, right? So we know a little bit more about which country, what specific little elements that they need, yeah, and their business environment. Okay. Some traps that you need to be very careful with, and you may already know this, the innovator have their strategies. So if you go upstream to plan, innovators, the reference product, they may have different strengths, different formulations, different polymorphs in different markets. When you're selecting a reference product, you need to be very careful, right? Because you may have developed your R&D pipeline based on European source reference product, for example, right? And then your marketing team says that, oh, we are going to sell the product in ASEAN. You purchase an ASEAN comparator and you do a BE study, you find that you failed. You wondered what happened, all right? What could have happened is that the reference product from ASEAN and the reference product from Europe is entirely different, right? They may have different formulation, different polymorphs, and you are caught by surprise, okay? So be very careful on when you're doing upstream development. I'm going to skip all these because you're going to have the slides anyway. We'll open a bit more for questions. Yeah. But those are the slides of what we can do here in Malaysia. We're hoping that we can do it together with IBBPS. Okay. Shaida, that's the end of my presentation. If there are already some questions in the chat box, or do you think that there are any questions, I'd be happy to answer them.
Thank you so much, Mr. Ken, for that much highly informative, detailed, and interesting presentation. So we are now moving towards the question and answer sessions. We have few of the questions in the chat box. I'm going to ask you questions one by one. So the first question is it mandatory to include both male and female subjects in bioequivalence study? And under what circumstances can a study be conducted using only one gender? Like, are we able to include only male participants or only female participants in bioequivalence study?
Okay. Thank you for the question. At the moment, ICH M13 says that it's recommended to have both genders. Okay, the exceptions are whereby if the study is not safe. The second criteria is always safety, right? For instance, if the product is a oral contraceptive, for example, you're doing a bioequivalence study on oral contraceptives, you will not do the study on male gender because you will not be able to assess correctly, right? Similarly, a classical example, if you're doing a bioequivalence study on PDE5 inhibitors, right, whether it's sildenafil, tadalafil, vardenafil, right, you shouldn't be doing this study on female subjects on the reason of safety, right? Because you will not be able to collect adverse events which may occur in the male gender, right? So in such studies, you will do an all-male study, right? In oral contraceptives, you will do an all-female study. So it depends on the product, and decisions are made based on those. Otherwise, if it's a regular product, can be used in both genders. The guidelines currently says that you should have a representation of male and female.
Thank you, Mr. Kent. The second question is, when changing an API manufacturer after product approval, what scientific and regulatory factors are considered to determine whether the existing bioequivalence data remain valid and a new B study can be avoided?
Okay. So API is something that you need to look into the particular country's regulation, right? It's driven by the variation applications. Okay. The guideline which we'll look at will be scale up post-approval changes, SUPAC guidelines. So if the API changes that or manufacturer are minor, which doesn't fall under the SUPAC class, right? You can actually support the biowaiver applications in terms of dissolutions and all. Otherwise, if the change exceeds the SUPAC level, right, you may require a new bioequivalence study. But most of the time, changes in API manufacturer is allowed to biowaive. You do not need a new bio study.
Thank you. Another question is, if the coating material of an approved bioequivalent formulation is changed while the core formulation, manufacturing process, and dissolution profile remain comparable, then under what circumstances would regulators require a new bioequivalence study?
The question about coating would then again fall at the same category of SUPAC. Right? If the coating material has got an impact towards the absorption, right? That the coating material is not just there's a functional purpose for the coating material. Very likely you require a new bioequivalence study. Biowave is not possible. But if it's a non-functional coating, right? Non-functional coating, and then you're able to show matching in dissolution profiles, you do not need bioequivalence study. SUPAC guidelines that will tell you exactly what needs a bio study and what doesn't need a bio study.
Thank you. Another question is, if a client needs data on a specific population like African, Germans, while the B center is in Asia, so what guidelines should the B center will follow?
The great news is ICH M13A. All right. You see, bioequivalence is not driven by race or a country of ethnicity. But the M13A says that the bioequivalence can be done on any population.
Okay, thank you. Another question is, if the commercial batch size is significantly increased compared to the bioequivalence batch, then under what conditions can the existing B data can be considered applicable, and when would a new B study be required?
That question, there are two prongs to the question. Firstly, you need to first ensure that your bioequivalence batch size is at least 100,000 or 1/10 of a commercial scale. All right. So let's assume that it was 100,000 batch size, and now currently you want to increase your commercial batch size to 1 million tablets, for example. You have to look at the SUPAC guidelines, right? If the change in your commercial batch size is significant, and there are certain process changes, or perhaps your excipient ratios may change a little bit, right? There are tolerance ranges. So if you exceed those tolerance ranges, the SUPAC guidelines would then ask you to do a new bioequivalence study. If it falls within those acceptance criteria, you can again go by biowave approach.
So we have another question, are there any criteria for blood group selection for B studies? If yes, please give scientific justification. Also, if no, please also give scientific justification. Is there any requirement for specific blood group enrollment in the B studies?
We always fall back on guidelines. Okay, it's always the best thing whenever you have uncertainty, you fall back on guidelines. So guidelines basically just say healthy volunteers, right? And it doesn't define a healthy volunteer. It doesn't say that group A is healthier than the group B or group O or group AB. It doesn't say that at all. All right. So the idea is that it has to represent the population, and in the population we have different blood groups also. It's almost, you will never find a protocol with a inclusion or exclusion criteria which specifically screen for blood group type. Okay, it isn't. However, having said that, there may be reasons that you may want to do pharmacogenomic screening for certain products which are known to be metabolized by certain pathways. Right? They are known to be slow metabolizers, fast metabolizers, but it's a little tricky because if you're going to do that screening, you're going to incur a lot of additional cost. Of course, it may give you better outcomes, right? If you have a more homogeneous, the idea, I think the question you brought up is that should we have a more homogeneous population, outcomes will be better, which is true. But if you look at it from the other end, if I have a very heterogeneous population and I'm able to pass bioequivalence, what does it tell you? You have a great product, right? If you're doing a bioequivalence study in a very heterogeneous population and your statistics is good, it only tells you that you've got a great product.
One last question for you. What is the best way to decide the study design, either it's 2-period or 4-period? Should we follow the FDA product-specific draft guidance? As in some cases, FDA recommends 2-period, but the available literature shows that the product is highly variable. So which design should we follow, either 2-period or 4-period?
I think this question would be driving towards the highly variable products, which it would be will be talked about in M13C. All right. How do you treat highly variable products? So the current thought process, ICH M13C is not out yet, but currently how agencies are viewing highly variable products is that they allow reference scale bioequivalence. So should you want to use reference scale bioequivalence statistics, this is your choice. If you want to use reference scale bioequivalence statistics, you will have to do a replicate design, i.e. 4-period. Okay. But if you do not want to use that statistics, you want to use conventional NOI statistics, then you can use a 2-period design, but your sample size may be increased much higher. For example, a replicate design 4-period, you may be doing 36 subjects, full replicate design, right? Should you want to fall back on a 2-period design, you can, but you may need maybe 70 subjects for the same project. So you do not have a guideline which tells you that you must do 2-period or 4-period. The question lies down would be that, do you want to use reference scale bioequivalence statistics? If you say yes, then you have to use the replicate design. If you say no, then you can use a conventional 2-way crossover design.
Thank you, Ken. I'm asking one more question from you. How do you select the reference listed drug or comparator product if not available in Pakistan?
The question here you should ask is that where you want to market your product. Okay, so the decision to select a reference product would be the decision where the market authorization is being sought after. Okay, even in the ICH M13A, right, it now defines, it doesn't use the word reference product anymore, it uses the word comparator product, right? And the comparator product's definition will be in the market which is being sold. Okay, so you have to decide, you say, oh, I want to do this project for European countries, right? So you need to source for European source. If you want to do this project to seek market authorization in ASEAN countries, for example, then you look for the ASEAN comparator.
Thank you, Mr. Ken, for sharing your expertise and addressing our questions. So now, as we come towards the end for today's webinar, I would like to express my sincere gratitude to our speaker, Mr. 𝐊𝐞𝐲, for sharing his extensive expertise, practical experience, and valuable perspective on optimizing return on investment in bioequivalence studies. I would also like to thank all the participants for taking the time to join us today. and for your active engagement throughout the session. We hope that today's discussion has provided useful knowledge, practical recommendation that you can apply in your professional and research activities. So on behalf of the IBBPS, 𝐻𝐤 University of Health Sciences, thank you for being the part of this webinar. We look forward to welcome you to our future scientific and educational events. Thank you once again. And goodbye.
Thank you, and all the best to you all.
Thank you. Thank you. Thank you so much.